Good morning, students. In the previous class, we read about the structural organization of cell. Then plasma membrane. Plasma membrane, I told you, it is the outer covering of the cell. Then we discussed about the many functions of plasma membrane. Among that, transportation. So transport across the membrane by the process of diffusion. Today, we will just carry on with this topic. Transport across the membrane by osmosis. So what is the definition of osmosis? The movement of water molecules through selectively permeable membrane from high concentration to low concentration. This is called as osmosis. So in the diffusion, I told you the movement of solid, liquid, gas, any of them can takes place in the movement. But here the movement of water molecules. I told you in a diffusion that essentiality of the selectively permeable membrane is not required. But here it takes place through semi-permeable or selectively permeable membrane from high concentration to low concentration, or you can say along with the concentration gradient. Both is same thing, from high concentration to low concentration. So this is the definition of osmosis. So there are few examples of osmosis like in unicellular freshwater organisms, and most plant cells tend to gain water through osmosis, or you can say absorption of water by roots is done by the process of osmosis. Okay. So the process of osmosis can be seen in a cell placed in solution of different concentration. Now explaining when we are placing the cell in different type of concentration of the solution. First solution we will read about the hypotonic solution. What happens in hypotonic solution? See, this is a cell and this is the cell's external medium, or you can say the solution. So in hypotonic solution, the concentration of water content is more in external medium or the solution as compared as to water content inside the cell. So we can say the water content is more in external medium and low in internal medium or internal content of the cell. So as the cell tends to gain up water inside the cell, so the water starts moving inside the cell more, and as a result the cell will get swell up. Example, if you are putting the raisins in a container full of water, the raisin will get swell up. So that is done. That is we can see in the hypotonic solution. Next, what is isotonic solution? In isotonic solution, see, this is cell and this is the external medium. The concentration of water outside the cell and the concentration of water inside the cell is same. So the equal amount of water will leave the cell and the equal amount of water get inside the cell. So as a result, the size of the cell will remain same. And at last, in hypertonic solution, the water concentration is less in our external medium and the water concentration is more inside the cell. So the cell will lose its water and the cell tends to shrink up. As you can do in a salt water, when you are putting any cell in a salt water, that cell will get shrink up because the water content is less in outside medium as compared as to the inside medium. So this is how we can see when we are placing a cell in different type of solution. In hypotonic, the cell will gain water and it will swell up. In isotonic, the cell will remain same. In hypertonic, the cell will lose water and will get shrink.